What happened? Can you? I cannot see my photo. The photo part is missing. Amit, the photo part is missing. Please check. Please check. The video is not coming. You have to. Yeah, I cannot see the video. Amit, please check and highlight.
Sir, please enable the video part of.
It is enabled. Can you please?
Sir, enable the video part from your end.
From my end, it is enabled.
Yes,
Video is
yes.
enabled. Just a second, I'll check. Video is already enabled. Video is already coming. It shows video is there.
Here it is showing, sir, video off.
No, you focus on this आभा वाला स्क्रीन ना, आभा को पिन करो आप.
Yes, sir.
जिसमें विज एनोनिमस वाला आईडी है उसको पिन करो. उसका वीडियो कैमरा ऑन है.
Yes, sir.
The camera is on here. Should I leave and rejoin?
Yes, sir. Once rejoined in this user.
Okay. Leave webinar. I'll rejoin. Just a second. You have to change the name again.
Yes, sir.
I'm joining with Ava 21 Medical. Okay. You change the name from Ava to Nilesh.
Yes, sir.
Is the video stay muted? Join as panelist. Please enable the camera. Amit, please enable the camera.
Yes, sir.
Now it is coming.
Yes, sir. Yes, sir.
Now the camera is coming.
Yes, sir.
नहीं अभी भी नहीं आ रहा. कैमरा अभी भी नहीं आ रहा. आप इसको फोकस करो. इसके पहले आ रहा था, अभी क्यों नहीं आ रहा? अभी आपका स्क्रीन आ रहा है. Still not focused. You have to pin a different images. Camera. Wait. What I'll do is please pin the second user as. Why the camera is not coming? Previously when we tried it was there.
Yes, sir.
What is the problem? Make Dr. Nilesh
Sir,
as
can you
co-host.
please? Yes.
Make Dr. Nilesh as co-host.
Sir,
Camera
already
is on from here.
you
It is showing the video is on, but video is not getting captured. Why it is not getting captured? Enabled one exception. I don't know.
Sir, from my side, your all users are co-host.
But why the video is not coming? Previously it was coming. Should I log out from all the systems? I'll log out from all the systems one by one.
Okay, sir.
Leave. Unmute. Everything is okay now. Thank you very much for the support.
Yes.
And I'm really sorry for this problem, but sometimes you can't do anything about this. Really out of our hands a few times. So let's start. All right. So we are starting with the IBQ sprint for biochemistry, and I welcome all of you who are here today with me. Okay. So let's quickly take a look at some very useful and important images from biochemistry. I'll talk a little bit about the background of those images, so that when you see those images in the exam, you are pretty much comfortable handling those images, right? Let's start. Okay, so image one, you can see something in the lens. Now this is a fairly common type of question where you get the different types of cataract. In this case, the cataract is like you can say it is the appearance of a sunflower. This is the copper. This is the due to the copper poisoning. You can also get the oil drop cataract. Sometimes similar image will be there where it looks like a drop of oil is there within the lens. So that is in case of galactosemia. So images on the lens talking about diabetes are pretty common. You'll cover them in ophthalmology also. This one is showing the cataract due to the copper. Okay, another image related to eye, which is fairly common, and we have been seeing this coming in the exam again and again. And this one is related to one of the vitamin deficiencies. This is related to one of the vitamin deficiencies. Anyone can tell me what is this? What is this? Anyone Anyone can tell me what is this? Dr. Ulsau, Dr. Navid, if you can quickly tell me what is this? What are you looking on the screen? Okay. This is what is known as the bite spot, seen in the vitamin A deficiency pretty early in the course of the disease. We get to see the bite spot. Very good, wonderful, wonderful, wonderful. This is the bite spot. Okay. Now, another pretty common question that you get to see is this image showing some pigmentation around the neck. Okay, and then you get lots of different types of questions. A lot of different types of questions are there related to this image. Sometimes they'll ask you what are the presentations, sometimes they'll ask you what is the diagnosis, sometimes they'll ask you what is the cause for this presentation. So like that, multiple aspects of the question can be there. Anyone can tell me what we are looking at? Any idea what we are looking at? Anyone can tell me what we are looking at? Okay, I think this is slightly getting delayed. Okay. Okay, what are we looking at? Anyone can tell me what we are looking at? I'm very sorry for this camera view. Yeah. Okay, very good, very good, very good. This is the Casal collar or the Casal necklace, the Casal collar or the Casal necklace seen in the niacin deficiency. This is seen in case of the niacin deficiency. Okay, other similar presentations which can be there. See, you can have the presentation like this also. You can have presentation like this. You can have presentations like this also. You can see the pigmentation is not limited to the neck. We have the dermatitis all over the body, right? So you can have the pigmentation on the hands and even the face. So a lot of pigmentation is there in this, the Casal collar or necklace. A characteristic feature of pellagra, a very characteristic feature of pellagra. All right. Now, can anyone tell me what you are looking at? At this point, can anyone tell me what you are looking at? Any idea? What are we looking at? Any idea what we are looking at? Okay, so this is a case of Alkaptonuria. This is a case of Alkaptonuria. We are having technical issues again. This screen, my screen is frozen. Disconnect. Okay. Admin, I'll need you to enable this ID as host so that we can capture the image. I have to log out and I've joined in again. Please quickly make this video as host. Quickly now. Please make this ID as host. What happened?
Recording in progress.
Please make this, join as panelist and switch on the camera. Please enable video. I'm really sorry for this technical issue. Okay, so what you're seeing on screen, is the Alkaptonuria. Here you can see the gradual blackening in the mucosa, in the sclera in this case, in the skin and the cartilage in this case, in the ear. And another characteristic finding in this disorder is the blackening of urine on standing. So I assume you would not have seen this change in the skin pigmentation, the eye pigmentation, and that is why I'm showing you this picture, that this is how the changes occur over time. By the age of 30s and 40s, the pigmentation will start appearing in the eye and the various different places in the skin. This is what is known as ochronosis. This is what is known as ochronosis. Ochronosis is the characteristic feature of Alkaptonuria. Wonderful. I'm really sorry for the problem with the right. Okay, this is another very common image. Very frequently we'll get to see this image. Certain variations of this image can also be there, and based on this image they can ask you different things. They can ask you what is the diagnosis, they can ask you what is enzyme deficiency, they can give associated clinical picture, and then they can ask you different questions. They can ask you which enzymes are likely to be elevated in patients like this. Okay, so first and foremost, what you have to remember, that this, okay, another picture which you can see is this is another image which is commonly there in the exam. All right, so we are looking at the condition known as Gaucher's. In Gaucher's, the enzyme deficiency is the glucose cerebrosidase. All right, and very commonly in these patients you are likely to find elevated levels of ACE, angiotensin converting enzyme, TRAP, the tartrate resistant acid phosphatase, and the characteristic enzyme chitotriosidase. These are the three enzymes which are elevated simultaneously in a patients of Gaucher's. The description of these cells is mentioned as crumpled or wrinkled tissue paper appearance. The term which is used is crumpled or wrinkled tissue paper appearance. This is the presentation in the cells, and this cell itself is known as the Gaucher's cell. This cell is known as the Gaucher cell. This is a characteristic of Gaucher disease, but we don't consider it for making a diagnosis. It is not pathognomonic because we also have what is known as the pseudo Gaucher cell. The pseudo Gaucher cell, which is seen in the chronic myelogenous leukemia. So you should keep this point in mind. The pseudo Gaucher cell is something which will be seen in the chronic myelogenous leukemia. Appears like Gaucher cell, has a different composition. All right. When you're talking about cells, it is very important to show you this next image where you can see the different types of cells in the peripheral smear. There's the peripheral smear where you can see the different sides of cells. If you focus on this cell, this is having very large number of lobules. Okay, this is a hyper segmented neutrophils. Normally you have anywhere between 2 to 6. You're supposed to have 2 to 6 lobes, but sometimes you can have more than that, or you can have higher frequency of 5 or 6 lobes in the neutrophils. All of this is called the hyper segmented neutrophils. Along with that, you can see the large red blood cell. All right, so this is the characteristic presentation of megaloblastic anemia. Megaloblastic anemia, can be seen in two conditions, either benign or B12 deficiency. The B12 deficiency itself causes the functional benign deficiency. So essentially, the megaloblastic anemia is a feature of benign deficiency and not really a feature of B12 deficiency. B12 deficiency causes the functional benign deficiency by a phenomena known as the folate trap. I'm sure you already know this. Now, if B12 is the primary culprit, other features which you might find in addition to the megaloblastic anemia, you can have the atrophic glossitis, loss of papillae, atrophy of the parietal cells, achlorhydria, intrinsic factor reduction. These can be the causes. These can be the causes for B12 deficiency. And along with the megaloblastic anemia, you also find the demyelination of the posterior and lateral columns. This is known as the subacute combined degeneration of the cord, sometimes simply written as the SCD, SCD or SSCD, the subacute combined degeneration of the cord, along with the megaloblastic anemia. This together makes up the pernicious anemia, which is a feature of the B12 deficiency. All right. Moving to another image, this is another pretty common image we should be knowing about. This is talking about the intestinal cell. Here we are looking at the intestinal cell. All right, and you can see this is the luminal surface, the one which is facing the lumen of the gut, and this is the basolateral surface which is interacting with the blood vessel. Now, on the luminal surface, you find two receptors, SGLT1, which is the secondary active symporter, and the GLUT5, which is the facilitative transporter. All right. On the other hand, on the basolateral surface, you only have the GLUT2. Please note, not shown in the diagram, but on the basolateral surface, you also have the GLUT1. GLUT1 is a common transporter in all the tissues where it interacts with the blood. It takes up the glucose for the basal metabolism. So GLUT1 will be there in all the tissues. Please note that sodium potassium ATPase is shown in this diagram, but it is not a direct participant in the glucose transport. Okay, so if we talk about the transporters of glucose, please don't include sodium potassium ATPase. Sodium potassium ATPase is not a part of the glucose transport. It is performing its own function, which creates a gradient over here. This gradient is then used by the symporter, that is the SGLT. Okay, so various questions can be there. They can put a question mark that how the fructose is going to travel. So which transporter is acting here, which transporter is acting here. So these are the different questions coming from this image about which you have to be careful. Moving on, out of the blue, when you get this image and they ask you, okay, what are you looking at? What is this image? Anyone can tell me what is this image? Can anyone tell me what is this image? What do you call this? This is having a name which ends with the phrased diagram. So what type of diagram is this? What type of diagram is this? Can anyone tell me? What do you call this diagram? If you can make out, it is talking about acidosis, alkalosis, acute acidosis, chronic alkalosis, acute respiratory acidosis, chronic respiratory acidosis. What diagram is this? This diagram is known as the Davenport diagram. This diagram is known as the Davenport diagram. It can be used to quickly identify the acid-base disorders. Very quickly, it can be used to identify the acid-base disorders. So remember this, the Davenport diagram, which can be used as a tool to identify acid-base disorders by plotting the three values. One is the arterial blood pH. Second is the arterial plasma bicarbonate. And third is by plotting the arterial PCO2. By plotting these three parameters, you are able to quickly diagnose or identify the acid-base disorders on the Davenport diagram. Now, when you're talking about biochemistry and when we don't talk about the colored reactions which can come in your exam, then it's not really an image-based session from biochemistry, right? So look at this. You can see a purple violet ring at the junction of two liquids, which is the positive result, and a negative or a control result doesn't have a ring at the junction. Okay, the question tells you that is a child with type 1 diabetes mellitus brought to hospital in unconscious state. On examination of urine, this is what you find. Now there can be a lot of different questions here. I have told you what is this test, so I'm sure all of you know what is this test. Anyone can tell me what is this test? What test are we looking at? This test is also commonly known as the modified nitroprusside test. This is the modified nitroprusside test. Anyone can tell me what is this? Modified nitroprusside test is the other name for the Rothera's test. Modified nitroprusside test is the other name for the Rothera's test. Okay, so please keep in mind this will identify the ketones. How do the ketones? The beta hydroxybutyrate is not going to respond to the Rothera's test. So please note, I've mentioned beta hydroxybutyrate will not respond to the Rothera's test. The others meaning acetone and acetoacetate, these are the ones which will respond to the modified nitroprusside test. Okay, we can have a lot of other different colored reactions which we get to see when you're examining the urine. For example, you can have the Molish test, which looks almost like the Rothera's test. See here, the purple violet ring is here also. In the Molish test also, we get the purple violet ring. For Molish test, what you have to remember, the Molish test is for any and every carbohydrate. Whereas the Rothera's test, I just told you, is for the ketones. Okay. Here we make use of the concentrated H2SO4 as the analyte. And in this case, you are making use of the ammonium hydroxide. Okay, liquid ammonium hydroxide is being used here to make the ring. Other reactions talking about the same type of compounds can be the OSA zone crystals. Again, talking about the carbohydrates, the OSA zone crystals can be there, where you have the sunflower shaped crystals for maltose. You can have the pincushion shaped crystals for lactose. These are also sometimes described as hedgehog shaped, the pincushion shaped, or you can also call it as a cotton ball shaped. These are the different terms used to describe the crystals which are formed with lactose. Okay, so very commonly we need to distinguish between the lactose and the maltose, and this is how we are going to identify them. The ozone crystals can be formed by the monosaccharides also. In case of monosaccharides, commonly you get to see the needle-shaped crystals for glucose, for mannose, and even for fructose. All of these are going to give the needle-shaped crystals. On the other hand, if you talk about galactose, galactose will give you the envelope-shaped crystals. Okay, envelope-shaped crystals will be a feature of galactose. So these are the ozone crystals obtained by treating the glucose containing solution with picric acid. So this is the reaction and the crystals which are formed. Very common image which will land up in your exam again and again and again. This is the Bendick test, commonly used for identifying the reducing sugars, but please note this will give a positive result with any reducing substance. You can get the different types of colors depending on the concentration of the reducing sugar. You can get different types of colors, and more the closer to red you are, the higher is the concentration. So we start with the blue solution. This is the Benedict solution. Then gradually it becomes green, then it becomes yellow, then it becomes orange, then it becomes brick red. Okay. So you can get question like this where you get multiple colored reactions and then you are given a clinical scenario and then you'll be asked which of the test might be positive. So till now I've shown you this is the Rothera's test for ketones. This is the Benedict test for reducing sugars, and you can find out, you can see that we're talking about a patient who's having the starvation. In starvation, only the ketones will be there. Okay, so in questions like this, you have to identify the condition, the scenario, look at the tests, and then mark the correct answer. The one shown here is for protein. This is the Biuret test. Okay, this is the Biuret test. Commonly it is used for the serum plasma estimation, but you can use it for urine plasma also. Okay, now I come to one very important set of images. I'll count this as one image, but this is actually a set of images where we look at the draw order for specimen tubes, which you commonly call as the Vacutainers. Okay, so the first tube, which is used when you are taking the samples, see there is a specific sequence in which the tubes have to be used when you are taking the samples for sending them to the laboratory. Okay, so this is known as the order of draw. The first one is for culture studies. The first tube is always for the culture studies, followed by the tube for coagulation studies. The light blue cap having the sodium citrate as the analyte is used for coagulation studies. When you want to estimate the INR, PT, APTT, for these you are going to use the blue top cap which contains the sodium citrate, followed by either the red top cap, red cap top, or you can have the yellow top. Both will have the same serum determination in biochemistry. You can see the red one here, there's no gel, whereas in the gold top you have the gel. This will also give you serum determination for the biochemistry. Now, some parameters will require plasma for estimation, and for this you can have the heparin in the tube, which is light green, or it can be dark green. The light green you have the gel, and dark green the gel is not there. When you want to do the determinations in plasma, certain estimations might be done in plasma, so that is where you get the heparin containing tube. Then you have this lavender top tube, which contains the EDTA, commonly used for the whole blood hematology, the complete blood count. For that, we require the intact cells, and therefore we must not allow the blood to coagulate. So, containing EDTA, you have the lavender top tube. The lavender top tube is exactly similar to the pink top tube, only difference is in the label. So you have special cross-match label for patient information on the pink top tube. Otherwise, the lavender top and the pink top tubes are exactly the same. They are actually the same tube, only the label is different. And then you have the gray top tube, which contains the sodium fluoride to block the glycolysis, and therefore it is used for the determination of glucose in the blood or plasma. Okay, so remember the gray top tube containing the fluoride, which is used for determination of glucose. So order of draw is very common, very important, and very frequently lands up in your exam. They can give you different tubes and they can ask you what is the analyte, they can ask you for what purpose it is being used. They can give you a combination of tubes, they can ask you which is the correct order for the draw. So like that, the different permutation combination question can be there. Okay, Some I told you dark green tube where the gel is not there, again for plasma termination. And the last tube to be used, if at all you're making use of this tube, this should always be the last tube. This is for trace elements, for toxicology studies, and for the nutritional deficiencies. For these purposes, you take the sample in the dark blue cap tube. Okay, so this is the order of draw which you have to keep in mind. Now again, very commonly you can get this graph, and there can be different permutation combination about this graph. Different questions can be there. Maybe sometimes they can give another graph with this, and they can ask you the type of inhibition which is seen in this particular image. So this graph is actually the line Weaver-Burke plot, also commonly known as the double reciprocal plot, also commonly known as the double reciprocal plot. All right, and the different types of inhibition which we can infer from this graph, you can see here. You can have the competitive inhibition, where the point on the y-axis is the same. This is the uninhibited graph, and this is the inhibition reaction showing inhibition. So the point on the y-axis is the same. You can have the non-counter inhibition where the point on the x-axis is the same. Okay, so in this case the point on the x is the same. Or you can have the uncounted inhibition where the two lines continue to run parallel, they don't share a common point on the graph. Competitive inhibition is very, very common. This is the mechanism which is also followed by a large number of drugs. So you should keep in mind when you're reading the drugs whether you're talking about competitive inhibition or something else. Okay, for example, the ACE inhibitors, they are competitive inhibitors. Okay, so very, very commonly you will get the question on the competitive inhibition where you should be able to identify and interpret this graph. Okay, question you can see here showing some image, some bite marks, some chewing points, some chewed parts in the lip. Also you can see various combinations. This is what is known as the self mutilation. This is what is known as self mutilation, associated with also nephrolithiasis and mental retardation. All of this is a feature of one characteristic disorder known as the Lesch-Nyhan syndrome. In Lesch-Nyhan syndrome, the self-mutilation tendency presents as nibbling of lips and fingers, bite marks on the hands, and wherever the child is able to reach. Okay, wherever the child is able to reach, based on that there can be damage. So in these patients you will have the hyperuricemia. Hyperuricemia can present as gout, it can present as nephrolithiasis, it can present as tophi. You can have the mental retardation, you can have the dystonia, you can have very aggressive behavior, and obviously you can have the self mutilation tendency. These are very important features of the Lesch-Nyhan syndrome, very, very commonly and very frequently asked. Okay, so I can see Naved has given the correct answer for this. So I would request all of you to try and give the answers in the chat on whatever image you see on the screen. See, when you make the effort, when you try to participate, तो आपका recall better होता है. You are able to recall and retain it longer if you are participating. It doesn't matter whether your answer is right or wrong. That is not the idea. The idea is to participate. When you participate, when you give the answers, then you'll be able to recall it longer. Okay, another important question. You can see here a structure is shown. This is a purine ring. Okay, you know in the purine there are two rings. A part of the ring is shaded, and now the question is asking which is the donor for the part which is shaded. So basically what we are telling you that you should know the donors of the different atoms in the purine ring. What are the donors of the different atoms in the purine ring? Anyone can tell me the shaded part coming from where? Which of these four contributors are giving us the shaded part? Anyone? So this is coming from glycine. This is coming from glycine. See, I'll show you the different atoms and their sources. If you can see here, glycine is the donor for 2 carbon and 1 nitrogen. Wonderful. Naved is again given the answer. And the N1 is coming from aspartate, N1 is coming from aspartate, C2 is coming from formyl tetrahydrofolate, N3 and N9 come from the amide nitrogen of glutamine. This is not the amino nitrogen, this is the amide nitrogen, the side chain. The carbon 6 is coming from respiratory CO2. This is something which is very frequently asked in the exam. Then the C8 is again coming from tetrahydrofolate, the methinyl tetrahydrofolate, but nevertheless it is the tetrahydrofolate. So this is an important image which you should remember. At the same time, you can look at the pyrimidine ring also, where we start with N1, C2, N3, C4, C5, and C6. Okay, here the C2 is still coming from respiratory CO2. The N3 is coming from glutamine, but the rest of it comes from aspartate. So another common question is, which amino acid is not contributing to both purine and pyrimidine? So you can see glycine is contributing to purine but not to pyrimidine. On the other hand, the aspartate and the glutamine are common to both. The glutamine and aspartate are common to both purine and the pyrimidine. They're common to both purine and the pyrimidine. So comparison purine and pyrimidine, and very commonly you'll get this question, which amino acid is contributing to both, which amino acid is not contributing to both. So like that the question will be there. So you have to keep in mind the name of glycine. Okay, this is a technique based image. If anyone can tell me what is happening here, see, this is what is known as the two-dimensional electroforces. The name is self-explanatory. When I say two-dimensional electroforces, you're doing the electroforces in two dimensions using two different techniques. We are performing the electroforces in two dimensions. The question asking, which are the two techniques which have been used here? The two techniques which have been used here for the two-dimensional electroforces, commonly written as 2DE. The 2DE includes isoelectric focusing followed by the SDS page. Isoelectric focusing followed by the SDS page. All right. So I'll quickly show you what I mean by isoelectric focusing and SDS page. So first, we separated the proteins on the basis of the change in their isoelectric pH, which is known as PI. So in this cylinder, we have separated them. Then we use this cylinder for a second round of electroforces using the SDS page. So now we perform the SDS page, and there is second round of separation, which is 90-degree to the original direction of separation. That is why we call it two dimension. First you are separating on one axis, then you are separating along another axis. This is called the two-dimensional electroforces. This is what we call as the two-dimensional electroforces. So first you have the isoelectric focusing using the isoelectric pH, and then you have the SDS page, which separates the proteins on the basis of their mass. Okay, so you should be able to identify this. Nowadays we are not doing IF so much, rather we go to Western blotting, or if the lab is very well equipped, then you can have the GCMS or MSMS for separation of the proteins. But those places which have low infrastructure, the two-dimensional electroforce is still a very, very important tool for separation of the proteins, for studying the expression of the proteins. Very, very important tool in the past also. Now you look at microRNA. MicroRNA is another very, very common, very frequently repeated question which is coming in the exam, and different aspects can be asked. In this case, we are talking about which of the proteins and enzymes are involved in the processing of the microRNA or in the synthesis of the risk. Okay, so here what you can see, one enzyme for the immediate modification. Okay, so this is the initial structure, which is called the pre-microRNA, and then two enzymes will quickly act to remove the cap and the tail. Then another enzyme is acting to transport it outside the nucleus, and lastly one enzyme is acting in the cytoplasm to remove the you can say the cap, to remove the cap or the folding or the loop from the RNA. So I'll quickly show you the names. You can see here. First, we have the drosha and DGCR8. I'll write the names here separately. First, you have the drosha, and the second enzyme is DGCR8. Please note, DGCR8 is also known as PASHA. These two enzymes will remove the cap and the tail. Then you have the molecule called exportin 5. Exportin 5 will transport the microRNA from the nucleus into the cytoplasm. And lastly, you have the enzyme by the name of dicer. Dicer is going to cleave the loop on the RNA before it can be loaded into the RISC. So I'll repeat again, microRNA is a very important, very commonly asked question, and you should be knowing about the microRNA. Now, this is a classical beauty where you see a peak in the absorption spectra of any body fluid. It can be serum, it can be plasma, it can be urine, it can be fluid fluid, it can be synovial fluid, it can be CSF. You take any fluid and you will get the same pattern if the person is affected. So the question is, this unique absorption pattern is shown by which analyte? You can notice that a peak is obtained at about 400 to 410 nanometer. Anyone can tell me what are we looking at? The answer is listed here. You're looking at what is known as the Soret band. Soret band is characteristic for which condition? Anyone knows? The Soret band is a characteristic feature of which condition? Soret band is a characteristic feature of an attack of porphyria. And therefore, the correct answer here is the porphyrins. The correct answer here is the porphyrins. The porphyrins will show the Soret band, which is a peak of absorption about 400 to 410 nanometer in any of the body fluids during an attack of porphyria. In between the attacks, the individual is normal. There are no clinical features, and similarly, there is no Soret band in between the attacks. Wonderful, wonderful. Vaishnavan, Naved, all of you have told us that this is porphyria, and the correct answer is porphyrins. Wonderful, wonderful. So remember that ring structure is, so ring structure will absorb the excess light at around 400 to 410 nanometer. Talking about graphs, I'm giving you more graphs. This is a graph which is very frequently utilized in the laboratory, in the clinical laboratory, for estimation of the different analytes. The autoanalyzer is making use of this particular feature only. Okay, so what is A and what is B? You can see they are having a peak of absorption at 250 nanometer, and they have a second peak of absorption at approximately 340 nanometer for one of the forms. These two are related molecules. What is A and what is B? What is, see, A is having higher absorption at 250, but at the 340 nanometer it doesn't show a peak, whereas B is showing a peak absorption at 250 as well as the 340. Anyone can tell me what are A and B? What are A and B? Very common analyte used in the autoanalyzer for estimation of the analytes, part of the whole reaction series. A in this case is NAD plus, and B is the reduced form, that is NADH. So we induce a reaction whereby the NAD plus is converted to NADH. So if NADH form is there and you measure the absorption at 340 nanometer, you'll find that there is a rise in the absorption. More is the NADH, higher will be the absorption. And in this way, you can do the quantification for the analyte through which is undergoing the reaction whereby the NADH is generated. So very, very common technique for measuring the analytes. We club it to one of the reactions for that particular analyte, and we measure the spike in the NADH to get an idea of the concentration of the analyte. We have more graphs. Okay, one more graph. Look at this graph. Identify the protein A. You can see the protein B. Next protein already identified. This is the hemoglobin, and you can see the oxygen hemoglobin dissociation curve, percentage of saturation, and depending on the partial pressure of oxygen. So here you have to tell us what is A. What is A? Anyone can tell me what is A? If you want, I can give you the options. Oh, the answer is also written here. A in this case is myoglobin. I'm sure all of you know that A in this case is myoglobin. Please remember the cause for this difference in the graph between A and B is the hemoglobin is able to bind with 2,3 bisphosphoglycerate, which shifts the oxygen hemoglobin curve to the right. On the other hand, myoglobin doesn't bind to 2,3-BPG. In addition to that, the muscles do not produce the 2,3-BPG. Therefore, the curve, or you can say oxygen hemoglobin dissociation curve for myoglobin, is following a very particular straight pattern, whereas a favorable pattern is seen with hemoglobin because it is able to bind with 2,3-BPG, which shifts the oxygen hemoglobin dissociation curve to the right. More is the 2,3-BPG, more shifting to the right will be there. So it can be like this if you have high percentage of 2,3-PG, which occurs at high altitude, and it makes the absorption of oxygen possible even at low partial pressures. One more graph. Okay, one more graph. You can see different parameters are labeled here. On this scale we have time. You can see on this scale we have time, and on this scale we have the relative absorption, or the relative change is there. You can see we're starting with the blood glucose. 0 means the fed state. In the fed state, the blood glucose is pretty much high. Then it starts falling as the time proceeds in the last meal and stabilizes at a certain level. On the other hand, liver glycogen keeps dropping, and by 12 to 24 hours, it is completely exhausted. On the other hand, you can see the insulin levels are high in the fed state, but they will start falling and then follow a plateau phase, steady state level in the fasting state. And opposite is there true for glucagon. Glucagon is initially low in the fed state, gradually rises and reaches a peak plateau, maintains at that level. Corresponding to the glucose, please keep in mind these two parameters will follow the glucose. When glucose levels fall, insulin will fall. When glucose level falls, the glucagon will rise. Now, along with this, you find that plasma free fatty acid levels also rise. When plasma free fatty acid level will rise and they reach to the liver, they'll start getting converted into the ketone bodies. So you see, once the free fatty acids are reaching the liver, it takes some time. Then the levels start rising and they peak even beyond the level of the free fatty acids. Remember, the ketone bodies will also get elevated, but their rise starts after a certain gap when sufficient amount of the free fatty acid is already there in the blood, reaching to the liver, because ketone bodies can be supplied only by the liver. Okay, moving to the next question. Now, this is, I believe, from the blind spot. What is a Ramachandran plot? Does anyone know what is Ramachandran plot? Here you can see the psi angle, the phi angle. These are the angles which are being measured, and based on that, you have to make a diagnosis, or you have to tell where the Ramachandran plot is used. What do you predict using the Ramachandran plot? Can anyone tell me? Is it used for viral load in HIV infection? It is used for estimation of histopathological derangement in oral cancer, or is it used to describe the secondary structure of protein, or for the configuration of the carbohydrates? How many of you heard about the Ramachandran plot? No? You haven't heard about Ramachandran plot? See, Ramachandran plot is used to describe the possible secondary structures of the protein. Wonderful, wonderful. Shoaib has given us the correct answer. It is used to describe the secondary structures of protein. Here, the phi angle is between the alpha carbon and nitrogen. The phi angle is between alpha carbon and nitrogen, whereas the psi angle is between alpha carbon and the carbonyl carbon. Where the carboxylic group is there, that is the carbonyl carbon. Using these two angles, we can identify and predict the permissible combinations for the secondary structures, right? As you can see, alpha helix is there in the lower left hand of the plot. In the lower left hand of the plot, this is the alpha helix, and this combination is actually the beta pleated sheet on the upper left hand of the plot. Okay, so these are the different possibilities which are identified by the Ramachandran plot, and it helps our description of the protein easier or simpler. We can quickly identify the angles, and when we assemble them, then we can get to know the shape, secondary structure of the protein. Okay, now we come to the image of, I hope you know what is this. This is the DNA, bDNA. And here we are talking about the distance occupied by one turn from one curve to the second curve. What is the distance occupied by one turn? This distance occupied by one turn is also described as the pitch of DNA. So essentially, we are asking you, what is the pitch of the bDNA? What is the pitch of the bDNA? All right, what is the pitch for the bDNA? So please note, the pitch for the different types of DNA will be different. For bDNA, which is the most common form of DNA, the accepted answer is 34 Armstrong, containing 10 base pair per turn. It contains the 10 base pair per turn. On the other hand, if you're talking about the zDNA, then it will be 12 base pair per turn. If you talk about the aDNA, it will be 11 base pair per turn. On the other hand, if you talk about C, D, or E DNA, in that case, we have 8 base pair per turn as the pitch of the DNA. Last word, sometimes when you're talking about the bDNA, these values may not be there in the option. If these values are not there in the option, your second best answer is 36 Armstrong, having 10.5 base pair per turn. So please remember this value. The preferred answer is 34 Armstrong. 34 Armstrong is not there. Very good. In that case, you go for the 10.5 base pair of 36 Armstrong. So I'm so happy to see all of you have given the answer. Vaishnavan, Dr. Navajan, Shoaib, all of you have given the correct answer. Wonderful, wonderful. So nice to see. So talk about lots of images. This slide doesn't seem to end. Okay, see, this is one of the very pretty common questions which has been landing up in our NEET exam, the FMGE exam, the INICET exam. They always talk about this bond which is there between the nitrogenous bases, the bond between nitrogenous bases. In this case, they've labeled, they've shown using the arrow, this is the bond we are talking about. Sometimes they'll not give the image, they'll just write nature of bond between two nitrogenous bases in opposite strands of the double stranded DNA. Okay, so this is actually the hydrogen bond. There has been slight error with the formatting. The image is slightly shifted. So this is focusing on the hydrogen bond. Hydrogen bond is very important. It is the only non-covalent bond in the DNA, and therefore it can be easily broken, and this results in separation of the two strands. So denaturation is dependent on the cleavage of this hydrogen bond by heating. So this is very important. It is holding the two strands, but it also separates easily. So you can separate the two strands by breaking these weak bonds. They are very numerous, and that is why they're able to hold the two strands together. Thank you, Shoaib. Very good. The correct answer is the hydrogen bond. Wonderful. Now we come to this structure. First, how many of you have seen or you know about this structure? See, the question says, what are the components at the core of the structure labeled as A and B? The outer layer is labeled, you can see it contains the phospholipid, it contains the free cholesterol, it contains the peripheral protein, it contains the integral protein, and in the core, you have certain molecules. Can anyone tell me what are these molecules? What are these molecules? What is A? What is V? What is A? What is V? Okay. See, A is cholesterol ester, and B is the triacylglycerol or the triglyceride. The triacylglycerol or the triglyceride. Okay. So this is the structure of a lipoprotein, a schematic image for a lipoprotein. The outer layer is made up of amphipathic molecules. The outer layer is made up of the amphipathic molecules, whereas the core is made up of the non-polar molecules. Basically, the structure is made to transport the non-polar substances sitting in the core, because they cannot travel on their own in the blood. So we need a mechanism to transport. We need a mechanism to transport these non-polar molecules, the cholesterol ester and the triacylglycerols. This is where the lipoprotein comes into the picture. Lipoprotein is a specialized structure for the transport of lipids in the blood. All right. Okay. Moving on. Okay, we have already marked this answer that we are looking at the triacylglycerol and the cholesterol ester. Now, how many of you have seen this image which I'm showing in the question? Anyone has any idea about this image that you can see in the question? See, there are two images here. One is a primary image, and then there's a second follow-up image. Okay, so just for information, I'll tell you that the first image is the model which is being used for genetic engineering. This is a model which is used for genetic engineering, where we insert certain genes into the bacterial system, and then we isolate that bacteria, grow the bacteria in large numbers, so that we can get the product from the gene which we inserted into the bacteria. If the insertion is successful, which we can see in this image, then we call it the transformation. So you see, I'm showing you two different images. One is the model which is used, and second is the use of this model for the genetic engineering. This particular model is commonly known as the blue-white screen, very commonly used to identify and isolate those bacteria where the integration with the target gene has occurred in the plasmid. Okay, so this is the blue-white screen, where we use the model where alpha complementation is there using lac operon, and then we try to insert the gene in this lac Z fragment, resulting in the transformation. Okay, I think that was all the questions that I had for you today. I hope you are benefited with the images that I have shown you till now. And okay, so that was the last image that we had. Thank you very much for joining in. Thank you very much for participating actively and giving the answers. I hope these images will help you, because they are the ones which are likely to land up in your exam. And when these images come, you'll have an edge over the other students who not had a look at the images before the exam. So I'll sign off for now. Thank you very much, and have a great night. Goodbye.